Firstly, I would like to tell that I'm really honored that I can speak today among our distinguished guests. I'm going to tell you today about our results on social modulation of learning. But let me start by describing my relation to Jerzy Konorski. So scientifically, I'm a granddaughter of Jerzy Konorski, as you can see. As you could hear before, he coined very important terms like neuronal plasticity or conditioned reflexes type 2 we use in our studies. And what I would like to show you today is that the methods the researchers use in their studies are different, but the basic terms put forward by Jerzy Konorski are still the same. So the story started several years ago when I was a young PhD student. And together with Tomek Werka, my supervisor, we performed the experiments in which rats were trained in two-way avoidance training, so aversive kind of the training. And we kept rats in 4 or 5 per cage. So when we put back a rat to the cage, to the home cage, we noticed that other rats started to be really aroused. They followed one another, sniffed a lot, and made a lot of rearings, so kind of exploratory behaviors. And we started to think if it's possible to measure such between-subject transfer of emotional information. So we designed a model in which we kept rats in pairs, and one of them was subjected to fear conditioning. We called these rats demonstrators. Then we put this rat to the home cage, and it interacted with its partner. We called it observer. So we had two rats, demonstrator and observer rats. As a control groups, we used demonstrators that were exposed to the novel cage without any aversive stimulation in it, and home cage controls. Then we measured acoustic startle response and exploratory behaviors and activation of the amygdala. So we noticed that our observers paired with shocked demonstrators were emotionally aroused as they had increased acoustic startle response, and also the level of exploration was significantly higher comparing to control animals. Then we looked at the activation of the amygdala. The amygdala, as you could hear before, is a structure involved in processing and storing emotions. That's why we decided to look at the structure. We used CFOS as a marker of neuronal activation, and we measured it in different nuclei of the amygdala, because the structure is very heterogeneous, both morphologically and functionally. And here are the results for different nuclei. And to our surprise, here we have the results for the shocked demonstrators and observers paired with them. And to our surprise, we saw that the level of activation of the amygdala in the observers is really high. It's as high as in the demonstrators. And in the case of the central amygdala, it's even higher. So the general picture was like this. We had very high activation of the basolateral and medial amygdala in the demonstrators, and even higher activation of the central amygdala in the observers. So then we wondered if this activation of the central amygdala is related to behavioral response of the observers. Because, as I told you in this model I've just presented, the level of exploration was really high. So the observers were active. So we wondered what happens if the observers are, for example, freezing or are inactive. So we designed another model in which demonstrators and observers were in the same cage, but the demonstrators were subjected to fear conditioning, and the observers could hear and smell them, and the behavioral effects of such interaction is that the observers freeze almost all the time. This is the level of freezing of the demonstrators and observers, and you can see that it's very similar. And of course, exploration is measured here as number of readings or readings time. It's very low. So we had two models, one in which the level of exploration was really high, and another one in which exploration was low. So we compared the activity of the amygdala, and in the first model we obtained very similar results as before. I mean, the central amygdala was really active in the observers, but in the second model we didn't have activation of the amygdala at all. So then we asked the question, does socially transferred fear influence subsequent learning? Because it's known that emotion exerts quite long-lasting impact on brain states, affecting perception, learning, and memory. So we wanted to check if the interaction between our rats can influence subsequent learning. And we asked also the question if this is specific to the treatment of the demonstrator. Because, as you remember, our demonstrators were subjected to fear conditioning. As Steve nicely explained, fear conditioning results in freezing response. Like here, there is no escape from the cage, so what's expected is passive response, namely freezing. And if the information interchange between rats was specific to the training of the demonstrator, we would expect increased freezing in the observers in the subsequent learning, but not in another cage. This is the cage for active avoidance training, and it's here when the signal stimulus, usually a tone, is switched on. An animal is supposed to move to another compartment to avoid a food shock, which is usually switched on a bit later, usually something like 30 seconds later. So what is expected from the animal is active response. And in this type of the training, so the instrumental kind of the training, okay. Here. Sorry. Okay, so in this instrumental types of the training, we need an active response, and an animal has to overcome the initial freezing tendencies to effectively learn the response. So we assume that if this exchange of information here was specific to the demonstrator's training, we could expect a higher level of performance here in fear conditioning, but lower in active avoidance learning. So this is what we saw. So we started with fear conditioning. The animals had interaction just before fear conditioning, and we tested them on the next day. And what we had was significantly higher freezing response in the observers paired with shock demonstrators. And it was true both for familiar and unfamiliar animals. But in this active avoidance response, we also observed better performance in the observers paired with shock demonstrators. We had three measures of this response. Percentage of avoidance response, latency, so latency of the response, so the latency is shorter than 5 seconds, means avoidance response, and longer means escape, and inter-trial responses, which were measures of general arousal. As you can see, In red lines, we have observers paired with shocked demonstrators. They were significantly better, both in the first session of the training, so just they were exposed to this social interaction before this session, so this was acquisition, the effect on acquisition of the memory, but also on the next day when they were tested without any interaction. So we conclude that such interaction enhances both acquisition and retention of active avoidance response. So to conclude this part, we observed that social interaction with recently fear-conditioned partner enhances acoustic startle response and exploration and evokes strong activation of the amygdala. And that specifically activation of the central nucleus of the amygdala is associated rather with active than the passive response of the observer. And such interaction enhances both conditioned freezing and acquisition and retention of active avoidance response. Then Marta Mikosz came to our lab and she asked if there are sex differences in the effects of such social interaction. Because in humans, it's known that there are some differences between men and women in empathy. We decided to check how this is in our rats. And we compared the groups of males, females, and females in two phases of estrus cycle, in the estrus and estrus. And we checked brain activation, looking at the amygdala, prefrontal and insular cortices, and for active avoidance learning. And to our surprise, well, in males we got very similar results as before. So you can see the activation of the lateral and central amygdala in the observers. Again, the activation of the central amygdala was higher in the observers comparing to the demonstrators. But in females, we didn't get any differences between groups. Here are shocked demonstrators, observers paired with them, non-shocked demonstrators, and observers paired with them. So this was for the lateral and central parts of the amygdala. Then we looked at the cortex, and separately for the prefrontal and prelimbic and infralimbic parts of the prefrontal cortex. And in males, we had quite high level of activity, similar to in the observers, similar to what we saw in the demonstrators. But in females, again, nothing. And in the insular cortex, we had very similar, the very similar level in all groups. It was a bit surprising because we decided to look at the insular cortex as it's known that in humans it's involved in empathy and such in emotional transfer. So then we looked at the performance of animals after social interaction in active avoidance learning. And here, as I showed you before, there's a similar measure. This is latency of active avoidance response for males, the estrus and estrus females. And this is for the first day, so the day in which we had interaction just before the first session of the training. And here is testing for memory retention on the next day, so the session of the second day. And in males, we got similar results as before. So the observers paired with the shocked demonstrators were better in both acquisition and retention of the memory. But in females, we had differences between the estrus and estrus females. The estrus females paired with shocked demonstrators were better on the first day, but not on the second day. So the retention of the memory was not affected by the interaction. And in the estrus female, it was the pattern was completely opposite. The observers paired with shocked demonstrators were worse on both the first and the second day of the testing. So what we can tell from this, this set of data is that social interaction with recently fear-conditioned partner results in different patterns of brain activation in males and females, and that in the US females it enhances acquisition but not retention of active avoidance response. So we hypothesized that activation of the central nucleus of the amygdala is associated with modulation of memory by social interaction with recently fear-conditioned partner. We are going to test this hypothesis in the near future, I hope. At the end, I would like to show you some new system of behavioral testing, which enables us to test animals and look at the social interaction in more ethologically relevant settings. So this is IntelliCage system, and it's possible to test a group of mice up to 10-12 mice per cage at the same time, and they are living in the cage, so we can observe their behavior continuously for days or even weeks. I would like to show you a short movie explaining how it works. So yeah. The animals are living in the big cage. There are 4 corners with bottles, 2 bottles per corner. The access to the bottle is barred by the doors. An animal can open it by nose poking, like this, and it can drink. Okay, what's measured is number of licks and visits and nose pokes. Here you can see it in transponders. To recognize single animals separately, every animal has a transponder injected under the skin, and when it enters the corner, it's recognized by the system. We can also apply air puffs, so this is negative stimulation, and as a reward we can put, for example, sweetened water to bottles. So I just want to show you 2 examples of applying the IntelliCages to the social experiments. In the first experiment, we wanted to see what happens if part of the group, in this case 2 mice out of 10, got aversive stimulation and how it affects behavior of others. And to do it, we firstly trained the animals in the place avoidance paradigm. So it means that they obtain air puffs in one of 4 corners. So they learn to avoid this corner. Here is percentage of visits in the corner with air puffs. So this is before the training and after the training, so they effectively learn to avoid this place. And then we extinguish this response just by turning off the air puffs here. So after 6 days, the animals started to visit this corner at the same level as before the training. And then 2 out of 10 animals was retrained, so they got the air puffs again in this corner. And we looked at the behavior of other animals, and it was in their visiting of this particular corner, but also other corners was inhibited by several hours following the treatment of this demonstrator mice. And it seems to be specific to the demonstrator mice present in the environment, because when we applied air puffs when no mouse was in the corner, but other mice, of course, could hear this air puffs, it was no effect. So we didn't observe inhibition of visiting the corners. And in the last, as a last example, I'd like to show you the training of APP transgenic mice. They are model of Alzheimer's disease, And they were tested in place preference paradigm, in which in one of 4 corners the animals obtain sweetened water. So they usually, normal mice quite quickly learn that it's a reward in one of the corners, and they visit this corner much more often than other corners. And we tested mice in 2 conditions. In the first condition, they were mixed, so the control mice and transgenic mice were mixed in one cage. And in other experiment, they were separated, so we had control and transgenic mice in different cages. And the results were a bit surprising, because in the group learning conditions, when they were separated, when the animals were separated, we compared the level of performance, so the percentage of visits in the rewarded corner on the first and the 5th day of the training for the wild type and transgenic mice, for young, middle-aged, and old mice. And as you can see, wild types acquired this response quite effectively. However, transgenic animals didn't learn at all. But when we put them to the same cage, so the wild types and transgenic mice were mixed, you can see that the transgenic started to learn. So they were able to localize the corner with the reward. So it's a proof of some social influence on learning. So this is the IntelliCage system is quite elastic, so it's possible to implement different behavioral design and to test social interaction in it. So I would like to thank all people involved in the experiments, the results of which I've just presented from my lab, especially to Marta Mikosz, who was involved in many experiments I presented, to Tomek Werka, Leszek Kaczmarek, Steve Maren, as well as Hans-Peter Lipp, who designed IntelliCages, and for a long time mentoring. Thank you very much.
One question that comes to mind is, did you look for the modality, the sensor modality that would actually be involved in transmitting the social transmission of the information? Or did you try to associate which modality it might be? Is it in particular interesting in case of Alzheimer? Is it just an auto cue versus a cognitive recovery by group housing them?
Yeah, it's a difficult question. I mean, it's not so easy to reliably check it, but we looked at this model of social interaction. We checked for modalities. We record ultravocalization, and what I can say is that not all animals vocalize. I mean, it's only like 30–40% of animals, so it's probably not the common way of exchanging information. It seems that more reliable, more probably is that it's olfactory, and in case of mice, it's also, I think that it's olfactory.
Thank you for an interesting lecture. I have a question dealing with the initial experiments. You have a demonstrating rat, and for fear, which is conditioned to near environment, the social interaction is also near environment. Does the animal who is a demonstrator extinguish fear faster when he has a contact with demonstrating with the other object that you demonstrate? Or does he maintain fear longer? What happens with the level of fear in the animal who is a demonstrator?
This is a good question. I mean, we didn't check it in demonstrators, but there are studies that show so-called social buffering effect. So you can expect, well, what they show is that interaction with non-shock partner allows for relieving fear faster. So such animals interacting with non-shock partner have lower levels of freezing as fear testing and extinguish faster. So, but we didn't look at it here. So it was focused on the observers.
Proportion of mirror cells in that situation, you know, affected by different states, hormonal states.
Well, this is, well, as you can talk about mirror neurons, to my knowledge, nobody showed mirror neurons in rats. Yeah, so it's hard to say if what we see in the amygdala are mirror neurons. But are you asking about the number of the activated cells in the amygdala depending on the hormonal status? Yeah, yeah. I think that it's that there is some influence on it. I mean, what we can see here is maybe there are different neuronal circuits activated because we look at it only quantitatively. I mean, it's just the number of activated cells. Maybe in females there are different circuits. I don't know.
Can I add more question?
Okay, go ahead.
Have you considered experiments to clarify which of the sensory systems are transferring information about the fear? For example, if blocking of the smell in a rat that is being demonstrated will block the effect of demonstration.
We didn't check it, but I know such studies which show that that olfactory cues are really important. So they were performed by blocking the olfaction in rats during social interaction. So yeah.
It's not my main field of interest, but as far as I remember, progesterone has kind of calming effect in women, that is reducing general reactivity. Maybe then diestrous females are more or less less reactive to anything.
Well, I don't think so, because we checked, for example, response to food shocks. Yeah, so and we didn't have difference. I mean, it was not directly, but we looked at the level of freezing, so the efficiency of fear conditioning in females, and we didn't have any differences here. So they clearly responded to food shocks at a similar level. Yeah, but.
Yes, that was averse conditioning, yes, and it was not really reaction in a demonstrator to observer. If it was in a female group, if it was the positive conditioning, maybe.
Yeah, maybe it would be different.
You didn't check. You didn't have a just.
No, well, it's in this experiments in the IntelliCages, cages, we often use females, and for this Alzheimer mice, it was females. So the effect is, and it was reward learning. So maybe you are right. You should check it systematically.